This media was gratuitously produced by Clear Contributions Audio Video. The intellectual property contained within belongs to Dr. Richard Gardner and Garland Science. Clear Contributions. Let's have clarity.
Let's talk about Hartwell's paper, and then Nurse's paper, and then Masui's paper. And then for Friday, remember, there's two other ones. So this homework was worth more because there's three papers, and that one's worth less because there's two. And that will end our homework for this semester. And we also have a quiz on Friday. It will be cell cycle stuff, obviously, and I will try to get together some kind of an email today in the midst of meeting with two groups and faculty meeting and all kinds of other stuff. All right, so Lee Hartwell. Lee Hartwell was a bigwig at the University of Washington in Seattle. He was like one of the original cell cycle. He's like one of the grandfathers of the cell cycle field. When I was a grad student at University of Arizona, we had the department. There was a molecular and cellular biology department had a seminar series. We had a seminar every week. We got some outside speaker to come in and fly in and give us a talk. The whole department was supposed to show up. All the grad students, all the faculty, everybody's supposed to show up. This was in addition to our journal club. We had like the journal club where one of us presented a new article and stuff. But we had a seminar. So someone comes in and gives a talk about their stuff, about their current research and so forth. And once every year, the graduate students got to get together and invite the speaker for one of those weeks. And so the graduate students all got together and we all voted on Lee Hartwell. So we sent him out this really nice invitation and had it all planned, and he accepted. And so we had this party planned and everything. We're going to take him out to dinner and have this party and do all this stuff with him, have this special reception. And this is our it's like a big honor, when the grad students all get together and invite you. And like 2 days before, he's like canceled it. And he said, sorry, but I'm too busy. And so what? Like, you're too busy? Well, yeah, you're busy. You're always busy. I mean, he's always a busy guy. So we what exactly is it? Well, he's just busy. That's it. That's all it was. Just busy. Well, it's like you knew you were going to be busy. We're all busy. I mean, everyone's busy. I mean, and that's great. So if you're too busy, then you shouldn't have accepted. I mean, we were just really ticked about it. And so I've never met Lee Hartwell. Now at the time, again, I told you my advisor, the guy in whose lab I was working, he knew Lee Hartwell because he was the postdoc in Lee Hartwell's lab when he discovered the whole checkpoint thing. And then when I went to University of Virginia, I worked in another guy's lab who was also a postdoc in Lee Hartwell's lab. So I have two of my mentors were both postdocs in Lee Hartwell's lab. So I call Lee Hartwell my sort of academic grandfather, even though I've never met him. And it turns out you can be a super, super smart person and still be kind of a flake. So and I shouldn't, that was just one time. So maybe that was just a bad week he had, but those things stick with you, All right, so, and then Lee got the Nobel Prize in 2001, I think, in physiology and medicine, for basically all this. So he's trying to study the cell cycle. What was his sort of paradigm? What was his another scientific example that he used to kind of base his idea on how to study the cell cycle? This is on the very first page here.
We wanted to use genetic mutants.
Okay, I want to use genetic mutants. What was this example that he had, or someone else had done, kind of genetic mutants to study something? It was the virus, the phage reproduction. So in these phage viruses, there's all these different genes. Each makes a different part of the virus. Each makes a different protein as part of the virus. And people had studied how this virus can kind of assemble and how it has to assemble in a certain order to get it right. And he's thinking, well, if the virus has to assemble in a certain order to get it all to be made in the correct way, the cell cycle has orders. It has to be things in the right order. And so maybe he can find genes that work in the right order to figure out the cell cycle. So that was kind of his inspiration for thinking about the cell cycle. So why did he decide to use yeast, other than it's just cool?
You use it because it's like a vitamin use.
Okay.
You can tell what cycle, what part of the cell cycle it's in based on the size.
All right, so that's one of the big reasons. Anything else?
Electronically, it's either haploid or diploid.
Okay, and that kind of leads into the other one, which is it's very genetically useful. You can do mammalian cells, like it says here, genetic studies with mammalian cells are impossible. Yeast, you can do genetics with it. You can mate an A and an alpha cell, and they'll form a diploid, and you can get meiosis, and basically you can cross genes just like you can in mice. And you can cross these and get babies and see where the genes are and how they're linked together and map it. And you can do all this in yeast as well, because the cells will mate and they'll do meiosis, and you'll get babies out of it. And like I said, you can grow them at haploid or a diploid. If you grow a cell as a haploid and it's got a mutation, well, that mutation becomes visible. If just going back to little genetics, if you're a diploid and you're big A, little a, and big A is dominant to little a, you see the big A phenotype. You don't see the little a phenotype because that's dominant. So you might have a mutation, but you can't see it because you've just got the normal phenotype. And only if you're little a, can you see the mutation. But if you're a haploid cell, and then the cell can either be big A or little a, and then if you got the mutation, well, you see the phenotype. It's much easier to work with. You don't have to worry about it being hidden by a dominant gene that's in there. So haploid cells can be easier to see a mutation going on. So they made them useful. And then like Danny said, they have the little buds. So you can look at a cell, and that cell is probably in G1, and then as it gets a little bud, now it's in S. Now it gets a bigger bud, it's in late S. It gets even a bigger bud, it's probably in G2. Or it could be in mitosis. So if it's got one nucleus, it's still in G2, but if they've already separated, they're probably gone through anaphase already. So G2 or M. So you can look at the cells, and that was really useful. And so he decided he's going to use budding yeast to figure out all these temperature-sensitive mutants. So then on the top of the third page, he isolated over 400 temperature-sensitive mutants. So this is a lot of work. I mean, this is the tedious part. I mean, you basically generally when you do this mutagenesis, you expose the cells to some mutagenic chemical, and then he would just simply have all these cells that have been exposed, and then raise the temperature up and see which ones don't grow anymore. You have to have a copy of the lower temperature. If you're going to kill your cells, you got to have some that are still alive. So you would have, for example, on your plate, you can put little colonies on your agar plate. So all these different colonies of cells, and then you can make a copy of that. And we haven't ever talked about replica plating, have we? Replica plating, it's a really low-tech technique. You got an agar plate, like you grow cells on, and you've got a little cylinder. It could be like a metal cylinder or a plastic cylinder that just fits the plate. So here's your little metal cylinder, and you got a piece of velvet that goes on top. So that's the velvet, and you can use a rubber band to get it all really tight. Or sometimes there's like another little device on here that kind of pulls the velvet really tight. And then you take your plate with the cells on it, and you just stamp it on the velvet. And now it leaves a little residue of each colony on the velvet. Most of the cells are still on the plate, but there's a little bit on the velvet. And then you take a clean plate that's never had anything growing on it, and you stamp that on the velvet, and you got the same kind of cells in the same kind of spot. So now you basically copy this plate, and you got the same. And these are just a few cells, but they'll grow up, and there'll be more of them. So now you've got two copies of all the same thing. So then you could take, as soon as you stamped it, you could take this and grow it at a high temperature. And if they're mutant, they won't grow. But if they're not mutant, they will grow. And then, so if you stamp all these, put it at a higher temperature, we're looking for temperature-sensitive mutants, and say, most of them are going to be fine. They're all going to grow. But maybe this one doesn't grow. So that one's temperature-sensitive. It's got a mutation. And then now you've killed your cells, but you still got the cells because you go back to the original plate. So now you've got your cells, and you haven't killed all these. Then you can test these again, and you've got them. So that's kind of what you could do with a replica plate. Just have a copy of your plate to do the test on, and then you've got your original cells. So he's going to find cells that don't grow, but then they might not grow for a whole bunch of any kind of reason that might have nothing to do with the cell cycle. So he gets 400 temperature-sensitive mutants that don't grow at the high temperature, that will grow at the low temperature, and then he's got to kind of look through all of them to see which ones actually all stop at the same place in the cell cycle. So once you found these, you've got to go back and retest them more carefully and look under the microscope to see if they're all stopping like this, or they're all stopping like this, or whatever. And so this is just a huge amount of tedious work. When you look for these, you might only get, if your plate has, a couple of hundred colonies of cells on here, maybe one of them doesn't grow, so you have to do hundreds and hundreds of these plates just to find a few of them, and to find 400 that didn't grow and then to sift through them all, that's just a lot of tediousness, which a lot of science is. So then figure 3 shows kind of what he's looking for. See, wild type cells, they're just kind of growing, they're just random places in the cell cycle. Some of them are at different phases at the higher temperature. So permissive temperature, that's the lower temperature. Restrictive temperature, that's the higher temperature. And the higher temperature, again, is where the mutation is supposed to kick in. We talked about this before, did we not? So it's probably looking for a protein that it's got a mutation where at the low temperature, that protein mutation doesn't really affect it, but at the higher temperature, the protein doesn't fold right and it falls apart and it doesn't function anymore. So the CDC mutant is what he's calling the cell division cycle mutant. They're all resting in the same spot in the cell cycle. So in this example, they all have big buds. But in a non-cell cycle mutant, they might all die, or they might even arrest and stop growing, but they're just stopping at all kinds of just random places. So that means maybe they couldn't make a certain nutrient or something like that, and they just sort of ran out at just random places, and then they just sort of stopped growing. So he's not interested in those, so we throw those out. Those are interesting to somebody else, but not to him. Keeps the ones that all stop at the same point, because that indicates, like, if they're all stopping in G2, it means that whatever gene got mutated, that's the gene you need to have to go from G2 back to the next, back into mitosis. And then, on the top of, I numbered these pages. I got this from a website that it had some funky software. You're supposed to read these things on the computer and not print them out. It's like and you click and see the next page and the next page. And I wanted to make a printable version, so I had to copy and paste everything and get it all formatted. And I forgot to put page numbers. But so 1, 2, 3 on page 4. We screened through hundreds of our TS mutants, temperature-sensitive mutants, discovered about 10% of them appeared to have a specific block in the cell cycle, so huge amount of work. And then down a little bit more, Joe Culodi, a graduate student, learned how to stain nuclei and analyze the nuclear morphologies as well. So to stain nuclei basically means to add DAPI. So remember, DAPI stains your DNA. So if you have before that, they could have a large cell, and you don't know if it's got one nucleus, and maybe it's not yet, maybe it's in metaphase, hasn't gone into anaphase, but if it's separated, now they've finished anaphase and it's actually split. You can't tell this unless you can stain the nuclei. If you just look at the cells, you can just see either way, just a big bud, but once you stain the nuclei, you can see. Yeast chromosomes, they're too small to really see the chromosome, but you can see a big blue blob. So this looks like a big blue blob. And I found it interesting when I read this that, this graduate student, learned how to do this. This is like new technique at the time. We're going to stain it with DAPI and you're going to see what it looks like. Whereas when I started grad school, everyone just did this. It was like, it's like looking back in history when someone first discovered, radio waves and wow, we could do, we just take it for granted now. DAPI is really easy to use. You just got this little bottle with a little dropper, and you have your microscope slide. The cells are on there. You just take a drop and stick it on the slide, put the cover slip, and there it is, and it just glows immediately. But this was a bigger deal to them because now they could distinguish that. And so then they narrow it all down and find that they actually found 35 different cell cycle genes that were controlling different, they're all separate genes, and then here's some of the mutants and what they look like at the higher temperature when the mutation kicks in. Let's look at figure 5. This is kind of cool. So here's where some of his CDC mutants worked. So on the right side, you got spindle pole body duplication. Again, the spindle pole is like the centrosome. It's like the microtubule organizing center. So that has to duplicate and separate. And then CDC4 has to happen in order for your spindle pole body to separate. And then CDC7, these are the proteins from these genes. That's required to initiate DNA synthesis. And then CDC8 and 21 are useful for DNA synthesis and so forth. And so these are where all these different CDC mutants worked on these sort of parts of the whole cell cycle. And there's a couple of different forks here because some of these things were separable. So bud emergence, CDC24 for bud emergence, meaning that if you didn't have CDC24, your bud wouldn't come out, but the rest of things would still happen anyway. So you could still like separate your chromosomes and do all this, but they just wouldn't be in a bud. So those are kind of separate events that happen at the same time. And so this was a huge amount of work after he got the mutants to kind of figure out where they all fit on this whole pathway of the cell cycle, which one did what. And even then, now we know we have a mutation where this doesn't happen, and we have to find a gene, an actual gene in the chromosome, and figure out what the protein actually does, how it actually works and so forth. All right, so this one special gene he found was CDC28. What does CDC28 actually turn out to be in hindsight?
CDK.
Yes, that is the cyclin-dependent kinase. So that is the CDK. So that was like the super important one because it's basically useful for so many different steps. You combine it with different cyclins and do different steps. So he found CDC28, and that was a big one. So during the time I was in grad school, we always talking about CDC28. And then the difficult part is if you're talking about fission yeast, they also discovered the cyclin-dependent kinase, and we'll just talk about that next. That was called CDC2 for their screen. They just randomly number their genes, and they just randomly gave it different numbers. And so CDC28 in budding yeast equals CDC2 in fission yeast, and that gets confusing. So nowadays the kind of trend is to when they discovered these, everyone just called it what they called it, CDC28 or CDC2, and it gets confusing. So more and more people are just calling it the cyclin-dependent kinase. But you still have to know, if you're in the field, you still got to know what the name of the gene is in that organism. Let's see. I was gonna make sure we're covering some of these. Let's see. So he goes and talks a bit about Tim Hunt and Paul Nurse. And here's Mark Kirshner coming up again. Remember Kirshner and Mitchison did the whole microtubule thing, but here's Mark Kirshner coming up studying MPF. And we'll come back to that later. Anyway, any other comments on Lee Hartwell's paper? All right, in that case, I feel like there was something else. Maybe it wasn't. All right, well, let's talk about Nurse's paper. All right, so Nurse I've never met. He's a British dude. I have seen him on a video, though. He's got this great accent, of course. And I don't know, it's about the same time. So Hartwell, early 70s, he was starting this stuff. And Nurse, I believe, is also in the 70s. And I don't know if Nurse had his idea independently of Hartwell, or I think he might have seen Hartwell doing this and decided to kind of do the same thing, but in fission yeast. So in his background here, he's talking about how other people at the same time were looking at the whole MPF in the frog eggs, and that comes up next in the Masui's paper. But everyone was kind of aware of everyone else's research, but no one at the beginning kind of knew how it all fit together. So that was kind of the big goal. We got Masui finding MPF, and nobody knew what it was, just that it was some factor that makes the cell go into mitosis. I like the way he puts down at the page 2 here, MPF resisted purification and molecular characterization for years after it was recognized as significant. So MPF, when they found it, all they knew was there was something in the cytoplasm of an egg you could suck out and stick into another egg, and it would make it go into mitosis. So they called it the maturation or the mitosis promotion factor, but nobody knew what it was, so they just had to make a name for it. But not a foggiest idea what kind of a molecule it even was. It was even a protein, it's a carbohydrate, nobody knew what it was. And for years, people were very frustrated by this, trying to purify it, trying to figure out what it is. They couldn't figure it out. So then at the same time, of course, Hartwell and Nurse were doing their screens, and the cool part about this is that Hartwell and Nurse, they were geneticists. So everything they was doing was all genetically looking for mutants, and the other guys were more sort of the traditional cell biologists, sucking stuff out of cytoplasm and injecting into another cytoplasm. Very different kind of research techniques, but they've all kind of come together. So Nurse is doing his fission yeast one. He finds a bunch of mutants that are also he calls them CDC as well, and he finds ones that have a Wee phenotype, again, small. The first of these mutants was located entirely by accident in Scotland, accounting for their name, during a visual screen for CDC mutants. So what made him especially attracted to these Wee mutants? Why were they more special than some of the other mutants?
Because apart from progressing in mitosis prematurely, it's basically the same.
Okay, so that was one of the big things. They progress prematurely into mitosis. So whereas a lot of the other CDCs would block you at a certain step, you would stop the cell cycle at a certain point and not go further than that. The Wee was kind of the opposite. This one, you actually went faster into mitosis. So it's kind of the opposite. That attracted their attention. And it kind of meant something was getting controlled here. You're actually you were turning on mitosis prematurely, as opposed to just like not being able to do a certain step. You were able to do all the steps and you were turning it on prematurely. So the normal Wee gene would be something that controlled when you went into mitosis. And so on the figure 3, kind of showing what that is. So again, remember that you have your cyclin-dependent kinase, and it must be it has that inhibitory phosphorylation. So Wee1 was what's putting on the inhibitory phosphorylation, and then CDC25 is the phosphatase that takes it off. So Wee1's the kinase. So if you don't have it, it's here, and this is what it's showing here, is that you have your CDK, which in this case is called CDC2. CDC2, CDC28 is the CDK. CDC25, the protein that gene makes is the phosphatase, and it takes off the inhibitory phosphatase. So removes this inhibitory phosphate, and then the Wee1 is the kinase that adds the phosphate. And it's an inhibitory phosphate. So by inhibiting it, this is the inhibition symbol. This is blocking this. It's stopping its function by adding this inhibitory phosphate. So in the Wee1 mutants, you don't have Wee1, so you're not adding that phosphate. So then this is active more than it should be. It's active before it ought to be active. And you don't need this to remove the phosphate because it never got put on. So then the cells go into mitosis prematurely. Does that make sense, everybody? The kind of the logic going on here? You're able to add that inhibition. So as soon as the cyclin starts to come up, that thing can just grab the cyclin, become active, and charge ahead into mitosis. So that's what they're kind of discovering there. And so the next cool part was they discovered that their CDC2 gene, which is the cyclin-dependent kinase gene, had a human homologue. Why is that exciting, first off? Yeah.
You mean that our bodies control cell division?
Yeah, and they do it just like yeast does. So, humans are not very much like yeast. I mean, we're very different than yeast. And if you can say, well, they could say, well, here's how yeast does it. We have this CDK and all that stuff. But, for all they knew, humans would be vastly different than that. Completely different way of doing things because, we're much different than yeast. But if they can show that humans have the very same gene, that suggests we're very related to yeast. That's a fantastic discovery. All right, so they actually showed that. How did they show that humans have a version of CDK, of CDC2, just like yeast does? This is this fantastic experiment.
So she went into the cDNA library and found what could possibly be a homolog, and then found temperature-sensitive mutations. That way, the human one would activate or would function at high temps, but yeast ones would not. And basically spliced it in there, and then they function, so it shows that it was the same.
All right, so she ends up getting a human gene that you can put into a yeast cell that's defective for CDC2, and it complements, it makes up for it, it does the same job. And this person was that a graduate student? Melanie. It is Melanie, I can't remember. Here she is here. Where does it talk about Melanie here? Trying to find a first reference. It doesn't say, it just says Melanie Lee in my laboratory. So I don't know if she was a graduate student or a postdoc or technician or whatever. Anyway, she works in a laboratory. She gets a human library, and just sort of transforms this yeast that's defective in CDC2 with this human library, looking for any gene in the humans that could like make up for it. And at the very bottom of this page, where the page with figure 5 and figure 4 on it, last paragraph, Melanie's rather bold approach required that the human gene be functionally equivalent to CDC2 rather than merely structurally similar. So it doesn't just have to have a similar sequence, it has to work the same way and be controlled in the same way. This was a demanding requirement because it meant that the human version of the protein would have to be capable of not only performing the same functions as the yeast version, but also responding to the same regulatory inputs. All right, so she finds something that works, but then when you find something that works in science, most of the time you get your hopes are dashed. You get all excited at first, and then it turns out that it doesn't really work out after all, and you got all excited for nothing. So that's why it says here, there followed a frantic flurry of experiments to ensure that the cDNA we had isolated really encoded a true human CDC2 homologue, gradually eliminated the possibilities that the cDNA had somehow been contaminated by the fission yeast CDC2 gene, or that we had cloned another gene that could somehow compensate. These controls seemed to go painfully slowly, but with the elimination of each alternative explanation, our excitement grew. Can you just feel it? I mean, it's just so stressful. You're going to go look at the experiment, and you're trying to rule out, you're trying to really be sure that this was it. And every experiment you do, you could find out that, oh, it was wrong in the first place. We just contaminated something, and we accidentally got a yeast CDC2 instead of a human CDC2 in there or whatever. And you try to eliminate these possibilities. Every time it gets closer and closer to finding that you really got the thing. But every time there's always a possibility that the next one's not going to work. It's kind of like if you're cheering for your basketball team in the NCAA tournament, and they win one, and then they're getting closer and closer, but every time they might lose the next one, until that last game, and you're just like, oh, this is it. This go all the way this far, and you don't want to. You wish you'd have just lost in the first round by now, because rather than having, get all excited for nothing, you get there, and so this is how they're feeling, and they're just, and it's just painfully slowly, you just can't, you gotta, and they finally, it worked, and it's like, wow, it's just like the hallelujah chorus comes down, and I mean, so I know what this feels like, and I know what it feels like to be disappointed by something that you thought was going to work and it didn't work out, and that happens more often than not. So they finally find this, and this is just a huge thing, and it shows that humans really are regulating cell cycle just like yeast does, and then they find out pretty much every eukaryotic organism is working the same way, and that was, no one knew that would be true, and so they finally go through here, and he talks in here about what was MPF, what does MPF actually turn out to be. We probably should talk about this in the next context, but he talks about it as well here. So, yes, it is a CDK cyclin complex. So MPF is, this is a big take-home message from a lot of this. MPF is the CDK and cyclin complex. And for all those years, no one knew what it was, but that's what it is. It's a CDK and the cyclin, which is the mitosis promotion factor. Let's see. And so he goes on and talks about how they kind of brought all this together. So he kind of discovered the CDK genetically, and so did Hartwell. And then on Friday, we'll talk about Hunt, who actually discovered the cyclins. But it all comes together because that's what the MPF is. I like the sort of second to last page here. Here's Nurse's pictures. The page before that, second to last paragraph, the picture that emerged from this work in late 1989. Now, 89. So now to put this in context, I started college in 85. And I came home from my mission, and I took a class in developmental biology about this time. And I remember learning about cyclins, and I didn't realize that this was something that had been discovered like in the last year. I just assumed people knew this for like the last 100 years or whatever. This was just not, and this is all was really new at the time. Late 89 was a single 2-component complex that controls entry into mitosis, meiosis. In a range of eukaryotes from fish and yeast through clams, starfish, xenopus, which is frogs, and humans. This conclusion brought about a satisfying convergence between genetic and biochemical approaches to the cell cycle control. Although at least some investigators had suspected early on that the two approaches might be detecting related activities, there was no obvious means by which to test the idea experimentally. This problem was solved once each approach had yielded a molecule. Yeast genetics identified CDC2, and biochemistry with MPF. And then the last little line here, with the cell cycle, as with many other cases, studies in model organisms paved the way to show that similar controls operated in human cells. It is probably safe to say that the problem would not have been solved without model systems that provided strategic experimental advantages. It is hard to imagine how it ever could have been solved using human cells alone. So obviously we want to know about human cells because we're humans, but by studying human cells, it would have been too hard to study. You can't do genetics on them. You can't do a lot of stuff on them. So you study it first in yeast, you find out how it works, and then you do a thing like Melanie did, and it works out and it shows. So just a great little advertisement for using model systems. When you're sending in your grant, you're writing a grant to fund your research, and everyone wants to know about human health and human cells, and your grant says you're studying yeast, and they come back and say, well, we're not interested in yeast. You say, well, yeah, of course we're not, but you have to agree with them, agree with an adversary quickly. But you're going to learn stuff in yeast you can't learn in humans, and then we're going to apply it to humans, and then we're going to go from that way. If you want me to study human cells only, we might not find everything. So that's the kind of goal on that one. All right, we still got more stuff to talk about. Let's talk about Masui's paper. So let's see, how do I find what we're looking for? How did Masui get into the whole cell cycle field? Because that's not kind of where he started.
Too difficult.
Yeah. So I got a kick out of this. He was studying frozen penguins, frozen penguin embryos. Someone goes to the Antarctic and mails you frozen penguin embryos, and he felt pessimistic about continuing to study cell differentiation. He wanted to study differentiation, embryogenesis, how cells differentiate and grow. He's kind of a developmental, how development works. And then it occurs to him that, some cell cycle events are kind of developmental, not quite as much as like a penguin embryo to an adult penguin, but this idea of the egg maturation, the egg has to kind of develop from its earlier form to its final form. There's a place where it kind of stops in G2, and then progesterone, the female hormone, comes in kind of every month or whatever and gets it going again, and then it gets kind of halfway through meiosis, and then it stops again until you fertilize, and then the sperm comes in and fertilizes. Then it actually finishes its second meiosis. So it's kind of this whole thing is called the egg maturation. It's like a developmental process. So you're like, well, this is kind of development. I like development. Let's kind of get into this. And so this is the guy that they start to do the injection experiments. And this is what we talked about before. You take a cell that's in like meiosis and inject the cytoplasm to another cell, and it drives it into mitosis. And so he called that MPF, for the maturation promotion factor or promoting factor. And then later on, turns out it drives cells into mitosis as well as meiosis. So now they call it mitosis promoting factor. So either one of those works. You should know, like on the test, if what MPF stands for and what CDC stands for, and some of these acronyms like that. All right, what was super surprising or amazing? They use this word amazing.
The cell cycle progressed in the
Yeah. So he says, amazingly, much of the cell cycle would occur in extracts. So you could basically, blend up your eggs and just get the cytoplasm and have this sort of goop, which is not even a cell anymore. And you could still add MPF, and the chromosomes inside would start to condense and act like they're going to go into mitosis, even though it wasn't in a real cell because you kind of broke the cells open. And it could go through 4 cell cycles just in these extracts. The chromosomes condense and decondense and so forth. You can't really do the whole, there's not really a cell for this thing to, cleave and all that, but it's kind of going through the process. What was really amazing was, anyone catch this part? They tried to purify the MPF, and they tried to break the cells open in 2 different ways, and one of them worked and one of them didn't. To investigate MPF's chemical nature, I attempted its extraction from frog eggs. This was unsuccessful until I found that MPF is quickly inactivated when eggs are homogenized, so you crush them up, but remains active in the liquid fraction when eggs are opened by crushing them with centrifugal force. You put them in the centrifuge and you spin them down, and that kind of crushes the eggs. The extra G's crush them, and that leaves your MPF active. But if you try to use a grinding tool to crush them, then it inactivates them. Just the fact he discovered that, I mean, it's like the most normal thing just to grind them up, and it doesn't work and doesn't work and doesn't work. Why would you even think to yourself, well, I'm going to try centrifuging them instead? I mean, that's just like, who would have thought of that? He just got lucky thinking of that, and it worked. And this reminds me of another story I heard, but it's a great science story. There was a lab that got a certain result. I can't remember what it was anymore. And they got it every time. And then another lab next door tried to replicate that result and could not get it to work over and over and over again. And they checked every single thing. They made all the new solutions. Everything you could possibly imagine they did, and they just could not get it to work. And the other guys got it to work every time, some biochemical thing. And after I don't know how long, they finally decided to actually switch laboratories. So this team in this room could get it to work, could not get it to work in this room, and this team couldn't get it to work, could get it to work in that room. And who would have thought of that, right? They would have just given up, but somehow they just got desperate, and the last thing I could think of was to switch rooms, and it worked. And it turns out this is back in the day when you could smoke in buildings, and the people in one lab were smokers, and there was residue in the air of cigarette smoke, and that inhibited their biochemical reactions, and the other room didn't have that. So I mean, just weird things like that you're just lucky to ever figure out. All right, so he does that and figures out MPF, but never quite. I don't think he himself ever figured out what MPF was, but it didn't happen until Nurse and Hartwell figured out the CDC2 thing, and they combine that with the cyclin studies and figure out the whole thing. Any other comments on any of these papers here? Hopefully I know we're all at a crunch time this semester, but hopefully these are fun to read because it's giving you kind of insight in these people's actual experiments. All right, let us move into continuing where we kind of left off at some point with cell cycle stuff. And so now, when you see this cyclin, oops, it's not going to work. Got to actually turn the screen on first. Come on, yeah. Screen mirroring, Durham 10. Oh, I can't. Oh no, not Durham 108. It's doing it again. That's what happened yesterday. There we go. All right. So when you see this, the cyclin-dependent kinase with Internet and Voodoo and Pandora under it, what is this? So what do we know this is now? This is MPF. This is the holy grail MPF, and this is what it actually is. So even when they knew there was cyclin-dependent kinase, they didn't know at first that was the same thing as the MPF the other groups had found. And they finally figured out, hey, what we found is the same thing as what they found. They called it this, we called it that, and it's all the same thing. So that was just way cool. Why is this not giving me a... How do I open this? Okay, I'm trying to figure out why I can't. Here we go, external monitor. All right. Now, we have talked about anaphase A and anaphase B. And just to kind of review anaphase A and anaphase B, let's find that again. All right. So again, anaphase A and B are simultaneous. Anaphase A, chromosomes, the constituent chromatids getting pulled apart. Anaphase B, the poles moving apart. On anaphase A, the microtubules are disintegrating, getting shorter and shorter, and motor proteins are kind of moving the chromosomes down as they get shorter. And then anaphase B, the poles are getting pushed apart, and there's motors between here that are pushing these apart. Now, think about, are these motors going to be kinesins or dyneins? First off, you got to remember which end of the microtubule these are. So which end's there and which end is out here?
Gamma would be inside the clusters.
All right, so you got microtubule this way and a microtubule this way. Is this going to be the plus end or the minus end? So these are the plus end. And now you've got motors, let's say a motor that's stuck to here and it's walking on here, and it wants to push them apart. Will this be walking toward the plus end or walking toward the minus end? It's going to walk toward the plus end. Let's say that's stuck on this one, it's walking on this one. It's going to try to walk toward the plus end. That's really going to push this way. And likewise, you have one here that's stuck on here, and it's going to walk toward the plus end. And that's going to push this one this way. So these will be kinesin proteins that are kind of in here pushing these microtubules apart. And then you've got some other microtubules on the ends here that are kind of attached to the cell membranes on the inside, and they're kind of pulling on those and kind of pulling the thing apart as well. So that's anaphase A and B going on. Let's see. Then here's one more issue that happens in this kind of, I should have put this first. This is kind of going back a step, but this is in prophase where the nucleus breaks down. Remember, it doesn't always break down in every organism like many yeast, but it does break down in most organisms. The nucleus breaks down. One way to break your nucleus down, or one of the important things you have to do, is break down the nuclear lamins. And that's like that cortex of proteins right under the nuclear membrane. And one of the important part of kind of breaking that down is phosphorylating those lamins. So here's all these phosphate groups getting added to those nuclear lamins, and that causes them to not stick together anymore, and they all kind of come apart, and that helps break your nucleus down. So if you're going to go into mitosis, and one thing you have to do is break your nucleus down, and to do that, you've got to phosphorylate those lamins. What enzyme might you think that might actually do the phosphorylation? It's a kinase, the CDK. So CDK does all kinds of stuff in the cell cycle. So there's a CDK is going to do that. And then now we come to a complex pathway of actually getting from metaphase into anaphase. And what it's going to be using is this blue molecule called the anaphase promoting complex. There's lots of different APCs, by the way, in biology. This one is the anaphase promoting complex. All right, so we already talked about the cohesins, which are these handcuff molecules that hold the two citric chromatids together, and they kind of wrap around and hold those together. We're going to basically cleave the cohesins so that the sisters can separate. To cleave the cohesins, that's the job of the separase protein. So the separase goes in there and cleaves the cohesins and allows these to separate, and then you've already got pulling forces. The motors are already kind of pulling on these things. There's already some tension going on, and once you cleave the cohesins and the separase does that, then they can separate. Now, you don't want your separase on prematurely, because if you prematurely cleave the cohesins and you do it before the spindle has been attached, then they're just going to float around loose in the cell, just floating around together, and then how are you going to make sure one goes one way and one goes the other way? So it's really important that you get these attached to the spindle so it's all ready to go, and then cleave the cohesins so that one goes one way and one goes the other way, so they don't just both randomly go wherever they're going to go. Does that make sense, everybody? So it's got to be done in the right order, and you cannot activate the separase prematurely before it's all set up and ready to go. Otherwise, they won't get pulled to right places, and they'll just float around, and they won't get wanted. And so that's the separase is controlled, and it's controlled because the securin holds on to it and keeps it from being active. So the securin is holding on to the separase, preventing it from activity. And then, in order to go into anaphase, the APC, the anaphase promoting complex, degrades the securin. NFase promoting complex is a bunch of proteases. It's sort of like the proteasome we've talked about, a bunch of proteases. So that cleaves the securin, which then liberates the separase. And then the separase can do its job by cleaving the cohesin, and then the things can separate. So it's multi-steps going on here. APC cleaves the securin. Securin then lets go of the separase. Separase then cleaves the cohesin, and then you go on. And it is the cyclin-dependent kinase that activates the APC. So add that to your list here. Cyclin-dependent kinase then activates this, which then cleaves that, which then liberates the separates, which then cleaves the cohesins, and then everything separates. So memorize this. This has got a lot of steps in it. So there are actually two things, and this gets you into anaphase, and then once you go through anaphase, you go to telophase, and then you have to exit mitosis entirely and go into G1. So to get your cell from metaphase into finish the mitosis, all the next steps, and then into G1, you've got to cleave two things. One is the securin, because once that cleaved, the separase does its job, cleaves the cohesins, allows anaphase. And then you've got to get the cells out of mitosis entirely, and that means you have to, and this goes back to this, then you've got to get rid of your cyclins. And the cyclins, again, are cleaved also by the APC, so the anaphase promoting complex. The anaphase promoting complex, okay, it's sort of the thing that turns on the proteasome or turns on the proteases. In this case, the APC is going to put the ubiquitin on, and that allows the protease to destroy it. So the APC is then kind of causing the destruction of the cyclins. But the APC itself is activated by the CDK. So the CDK will activate the APC, and the APC will do two things. First, it will cause the securin to get degraded, which allows anaphase to happen. And second, it causes the cyclin to be degraded. And once the cyclin's gone, the cell exits mitosis. So there's sort of two steps involved. Get rid of the securin and then get rid of the cyclin. And they have to go in that order. Securin gets you into anaphase, and then cyclin gets you into the next G1 once you destroy them. So you got a lot of destruction events going on. And I'll just mention for a last little thing here that before all this was discovered, people knew about proteases, and they were basically just used to like degrade stuff you didn't want anymore. Just garbage. It's like a garbage dump. You just protealyzed old proteins you didn't want. The whole idea of actually getting rid of something to control an event. We're going to get rid of cyclin so we can actually push the cell into G1. We're going to get rid of the separase so we can separate the chromatids. The control an event, that was a different idea of what proteases could do. So before this was all discovered, again, they were just meant to old stuff you want to recycle. They were kind of boring. And now suddenly they're exciting because they're actually making events happen. They're controlling the cell cycle. They're pushing from one stage to another by getting rid of cyclins or securins. All right, we are out of time. So on Friday we've got these other two papers, and you can tell they take a little while to talk about. We've got a quiz. I don't know if we'll have time to finish on Friday, so on Monday we can finish the whole cell cycle stuff, and then Wednesday and Friday we'll do 3 talks on one day and 3 talks the other day. Unless anyone really wants to get it over with on Monday. If any group wants to do it on Monday, we can do that. Otherwise, we'll do 3 and 3. All right, so then we also have, I think, 3 groups coming tomorrow. And I think did you ever get the emails I sent out? Was it yesterday? I sent out some notes, and they're kind of rough, but hopefully they help a little bit, and then we'll go over the stuff. So please remember your times.